going to be talking about personalized medicine through an assay that we've been working on developing, myeloma drug sensitivity testing, which we abbreviate as MyDST. And let me just say at the beginning, if there are any questions, feel free to interrupt me. I think about multiple myeloma all the time, so I tend to get down in the weeds. So if you want me to stop at any point and go back and explain something, or have just any questions, just please give me a holler, unmute yourself and give me a holler as I talk. So made a brief outline for what I'll be discussing today. First, I'm going to give you a little bit of kind of 3,000-foot view of multiple myeloma, some introduction, then jump in and talk to you about MyDST and the development of MyDST drug sensitivity testing. Tell you a specific story that we're about ready to submit about MyDST, the application of MyDST to improve the way we give monoclonal antibody treatments, our approach to that in multiple myeloma. And then lastly, just a couple of brief quick hits about current projects in the lab that we're working on, such as one that we're working on, MyDST for T-cell engaging therapies such as bispecific antibodies. All right, so I'll start just by kind of looking at blood cancers in general. This is the hematopoietic tree, the different cells that are derived from the hematopoietic stem cell that lives in our bone marrow. So multiple myeloma is a common lymphoid malignancy. So from the hematopoietic stem cell, you can get at the highest level leukemias that are derived from either the stem cell itself or from its progenitors that are either myeloid or lymphoid. And these more differentiated tumor types, lymphomas and multiple myelomas, are usually derived from, at least from the B, on the lymphoma side, B cells. One of the most differentiated cells that comes from B cell development is the plasma cell, which is our antibody producing cell in the body. And cancers that have the phenotype of that plasma cell we call multiple myeloma. So if you look at the overall, I guess, bubble graph of blood cancers, non-Hodgkin's lymphoma or lymphomas are probably the biggest bubble. Myeloma, so age is on the y-axis here, so it's a disease of older people. The median age is about 75 years of age, so it's a disease of older people. It rarely happens in people less than 50. The incidence is one of the larger ones of blood cancers. And the median survival rate, this is an old slide actually, has been kind of shifting to the right. Used to be that less than 40% survive more than 5 years. And this has really kind of shifted much more to the right. So the survival since the year 2000 really has improved quite a bit. Right now, we can tell patients if they're optimally treated and eligible for things like a stem cell transplant, an autologous stem cell transplant, their survival is expected to be greater than 10 years. So things have improved quite a lot, which is great. So the hallmark of multiple myeloma is that these cells share the feature of their ancestor, the cell that they developed from, the plasma cell. So they secrete antibody protein. Because it's derived from a clonal population, that antibody is also clonal. So we can distinguish this by lab tests. So we have a couple of different lab tests that we use as our tumor markers to diagnose the disease and then also to follow it over time. So our serum protein electrophoresis is just basically a Western protein gel in a Western blot where we can identify what usually should be a smear in the gamma region, shows this monoclonal population of antibody, which increases in the spectrum of monoclonal gammopathy of undetermined significance, which is a very common condition as you get older, benign form of myeloma. Smoldering myeloma is basically a pre-malignant transformation, and then multiple myeloma is basically more antibody is detectable. That antibody can cause problems, kidney failure, patients get lytic bone lesions, and so it can be quite debilitating if not controlled. The second assay that we have is this serum-free light chain antibody test. So basically, in your intact antibody, it's a Y-shaped protein. You have these heavy and light chains, so big and little parts of the protein some light chain normally circulates free for all of us, but it's a very low level. But people with multiple myeloma, they have much more antibodies, so they have quite a bit of circulating free light chain, and that's detectable by this immune assay. So both of these things we use regularly clinically in patients, and they get this test every month, both tests every month. So if you think about the genetic alterations that cause multiple myeloma, they're quite diverse, they're heterogeneous. They generally, this is kind of basically just telling you what causes multiple myeloma. So usually it's basically about a third of patients will have translocations of chromosomes that break and rejoin. And usually that comes from the heavy light chain loci, the genes that create antibody, that basically express antibody protein. They're highly active in these cells. So if they get translocate with acceptors such as cyclin D1, which puts cells through the cell cycle, then you have a cell that basically grows uncontrollably. So there are a diverse set of those, such as cyclin D1, FGF receptor 3, MYC translocations also occur in about 5% of patients. So there's all kinds of different translocations. People do get point mutations or deletions, such as loss of P53, the guardian of the genome, RAS mutations, RAF mutations, IDH mutations. So lots of different things could cause myeloma. The overlap, the ultimate result of all that is that these cells just grow more rapidly than they should, and they don't die when they should. So about a third of patients can be broken up into these different categories of either having translocations, gains and losses of chromosomes, deletions, or point mutations. So it's kind of a heterogeneous group of disease. Ultimately, the phenotype is this plasma cell that grows inappropriately and doesn't die when it should. So clinical problem that I'll kind of talk about addressing today with MyBST comes in two parts. So the first part is that we have a lot of different drugs that we use for this disease, which is a good thing, but on the other hand, there's no clinical test that really can inform the treatment decisions. There's no group of patients that genetically are going to respond better to one or the other drug. So we have a lot of options, but not kind of a lot of guidance to determine what to use. So the hallmarks of really our treatment in 2022 are proteasome inhibitors such as bortezomib or Velcade, carfilzomib or Kyprolis, immunomodulatory drugs such as thalidomide, lenalidomide, pomalidomide. We've always used steroids for this disease, so a potent anti-inflammatory or agent, dexamethasone is usually our steroid of choice for this disease. And these are used in combination, so steroids are basically always part of our regimen. We do use some old school chemotherapies, but it's getting more and more unusual. So alkylators such as cyclophosphamide, doxorubicin, anthracyclines, these are basically just kind of reserved for emergency situations now because of all the targeted agents we have available to us. Monoclonal antibodies, I'll talk a little bit in the middle of the talk. The most successful one has been daratumumab, which is an anti-CD38 agent that has been approved since 2015. We've got a new drug that came out last year, selinexor, which is a nuclear export inhibitor. And we've recently had FDA approval of some next generation immune therapies. We kind of abbreviate these Tcell, which is a CAR T-cell product, so chimeric antigen receptor T-cell product. Cilta-cel was actually just approved last week, a second CAR T-cell product, probably a superior one actually. So that's very good news for our patients. And there are several bispecific antibodies, which basically act also to harness the power of T cells against multiple myeloma cells, are looking very promising and probably will go out for the FDA later this year. So a lot of stuff. So part two of this is that these treatment decisions are really made without any prior data really, other than just the clinical experience of the physician who's treating the disease and how much myeloma they're used to seeing. And so it ends up being kind of a very complicated decision-making process where first so this is kind of a graph of disease burden, which is measured by antibody. When the patient's diagnosed, they get a multi-drug combination. This one is kind of an outdated regimen that's based on a proteasome inhibitor. That had a couple of bumps, so they went through some high-dose chemotherapy, had a treatment change to a stronger proteasome inhibitor, had an immunomodulatory drug added in. Then it went through a stem cell transplant. Didn't mean to advance there. And then kind of went through, just as you can see, multiple lines of relapse. They're on maintenance therapy for a while, started to relapse. Each time one of these peaks comes up, we need to change our recipe. So went through a lot of different treatments. And then you can see towards the end of their life, basically had a relapse that was not controlled and drug resistant. So very active disease at this point and many complications. Ultimately took this patient's life. Unfortunately, after 6 years with the disease. And so adding up all the drug costs that went into this patient's treatment, almost $3 million just as an estimate. So this extremely expensive disease to treat these days with all of this stuff we're doing. So it would be great if we could personalize people's regimens more than we're, start to personalize medicine for people with multiple myeloma. So the Multiple Myeloma Research Foundation is the largest foundation in this space. They've launched a clinical trial, it's called MyDrug. It's based on kind of the theory that the genetic alterations that patients go through may sensitize them to certain drugs. So you can see this point mutations that I mentioned in the beginning, such as RAF, RAS, IDH, the CDK pathway, FGF receptor 3, one of the translocations. If you have that identified, basically your patient will go to that line of therapy. And all of this is based on a backbone of getting a immunomodulatory drug, a proteasome inhibitor, and dexamethasone. So 4 drugs altogether. So it's personalized from the standpoint that they get an additional drug. So our approach is a little bit different. So we think of this, at least I think of this as more genotype drives proliferation, phenotype drives our drug sensitivity. So it's not necessarily the genotype that or treatments are geared towards, it's really the phenotype. So the phenotype I ultimately care about as a physician is my patient going to be drug sensitive to what I'm giving them, or are they going to be resistant to it? And should I pick another therapy? So I guess you would call that functional precision medicine, and that's kind of the dream in oncology, but has been hard to obtain. I think multiple myeloma is the perfect disease to break through in this space for this type of assay. And others have tried this too. So the competition basically is at Moffitt, WashU. There's a group in Finland and Mayo Clinic. So I will kind of go through the details of this table, but basically to tell you, everyone CD138 selects their cells. CD138 is a myeloma marker. They basically purify their myeloma cells through a magnetic bead column and then study their drug sensitivity in a dish, basically through various means, either a plate reader or a microscope-based technology. Our approach has been different than all of those in that we don't separate out the myeloma cells from their friends. We leave them in the context of the bone marrow microenvironment so that and the advantage of that is the viability is much better, as you'll see in a minute. And you can capture immune-related effects such as monoclonal antibody effects that happen through macrophages and NK cells. You can measure, as we found recently, effects that are mediated through T cells that are endogenous to that patient, which I think is a big advantage. So our readout here is flow cytometry. So jumping a little bit more to our technique specifically. So as I mentioned, we call this MyDST. So we obtain bone marrow aspirates from our patients, which is a standard clinical procedure to diagnose and follow the disease. We put it through a 96-well plate format in basically a 48-hour culture, so a short-term culture, and in the presence and absence of the different drugs, so untreated and drug-treated conditions. And we measure the effects of the different drugs that and their ability to reduce the viable myeloma cell populations from these patients using flow cytometry. So the goal is basically at 48 hours to get this report that that will show you what your patient's sensitive or resistance to. So a couple of things we've learned along the way. we picked 48 hours because basically you take a biopsy from a patient or you thaw their sample out from the freezer. They take a viability hit in the first 24 hours. Then they kind of settle in for 48 to 72 hours after that biopsy is obtained. And then they undergo a second phase of decline. So we found 48 hours. It's a short-term readout, but it allows us to basically have something to measure in our cultures. So we don't add any perturbations to keep them alive necessarily, other than some IL-6 to give them some growth cytokine. So we found, as I mentioned, that CD138 selection over a magnetic B column really is detrimental to their viability. So you can see that here, basically at 48 hours, have that hit in viability, but viability is much better if you use whole bone marrow cultures than in CD38. It's hard to measure a drug effect if you only have 10% of the cells surviving after you perturb them with this magnetic B column. We've also found that if you, when we used humanized media, which is the blue bar here, that cells have even better viability. So basically humanized, what I mean by that is that we try to replicate as best we can the plasma concentration or the serum concentration in the body, physiologic levels of amino acids and sugars and those things, so that they're not in a cell culture type media. They're more in a media that is conducive to that they're used to in the human body. So that's helpful as well. So our initial study on this, which we've published, is that we had a group of 55 patients. We basically looked at them from kind of the newly diagnosed setting, which is the left side of this graph here, this left side of this heat map. First relapse, so the first time they have relapsed their disease. And then with increasing lines of therapy, the multi-relapse setting, in this case, we studied people that had up to 6 prior lines of therapy. And you can kind of get the impression that at the beginning, there's some red, so some resistance. So red is resistant, green is sensitive. So a lot of green, not much red in the beginning. And then as you go to the right, more and more lines of therapy, more and more drug resistant, to the point that once they get to the 5, 6 lines of prior therapy, they're pretty multi-drug resistant, unfortunately. And that's why it's so hard to treat them in that scenario.
Hey, Dan.
So, yeah, go ahead.
We have a question that was posted in the chat, and it was how to differentiate direct anti-MMs versus immune stromal effects or tumor microenvironment effects.
Well, we basically have these in mononuclear cell cultures, so everything is there. I'll talk a lot more about this to kind of get to your question, I think, when we talk about the monoclonal antibodies, which is kind of the middle part of my talk. Because monoclonal antibodies require macrophages, they require complement. Our cultures don't have complement in them, but they do have NK cells and macrophages and T cells, all endogenous to that patient. And so we have done some experiments to kind of tease that out. Is it the NK cells that are doing this, or is the macrophages? Something that we're certainly interested in doing more, but basically the ultimate readout is the viability, and that could be direct tumor killing in the case of these drugs, like bortezomib, lenalidomide, the IMiDs, the immunomodulatory drugs, the proteasome inhibitors. Monoclonal antibodies is dependent on, and that's why none of those other competing assays were able to measure monoclonal antibodies, because they don't have that immune microenvironment. So yeah, I think that's a really important question. So we can see basically in this patients that we're recapitulating what we see clinically. At the beginning, they're quite drug sensitive, so a lower result in this graph is better, and they're getting drug resistant as we go to multiple prior lines of therapy for our key drugs, which are the proteasome inhibitors and immunomodulatory drugs. So kind of taking our ex vivo results in the lab and comparing them to how patients actually did in the clinic, we saw that this actually predicted how people did pretty well in terms of both their response and their event-free survival or progression-free survival, basically. So this is their response. So this is the newly diagnosed patients, where the patients received drugs that we'd actually measured in our assay. We can see that if we scored, and we kind of have this basically a 20% decrease in viability set is basically our cutoff point to say you have at least have to have 20% killing to be sensitive. So if you have no drug or one drug that were sensitive that the patient actually received, you can see that those patients didn't have a very deep response clinically. So they didn't, this is basically from 0%, so down indicates a response, 100% being a complete response, so that's ideal. But then if they got at least 2 drugs, they did pretty well. And you can see most of these people got to a complete response. A couple of them nearly what we call a very good partial response or 90% decrease. And then if they're sensitive to all 3 drugs they got, they did great. They all got to a complete response basically. And that's what we want to happen. So we are able to identify the problem patients here, or the problem drug treatment selections. In the relapse setting, similar results. Basically, the people that had 0 or 1 drugs that they were sensitive to in the assay didn't have good response, or they had progressive disease. Whereas the people that had at least 2 drugs they were sensitive to did better. And we looked at their events-free survival, basically when they had to change regimens or had a relapse. The people that were one drug sensitive or 0 to 1 drug sensitive in both the diagnosis and the relapse setting did worse. And those people that had at least 2 drugs they were sensitive to did better. So this is kind of what we would hope to achieve clinically. So we're hoping that application of this assay clinically, the ultimate goal would be be able to prevent inadequate responses in people by better drug treatment selection and extend their time in remission, which would be fantastic. We actually were able to score sensitivity and specificity. So basically your classic laboratory assay parameters. So in this graph, I'm showing you the clinical response on the y-axis again. So going down on this axis is good. You're responding at a complete response when you get to -100. And then the additive ex vivo combo effect is basically, so all our drug treatments are in singlet treatments, so not how we would treat the patient, but single drug treatments. And we add these up to an additive combo effect. It's basically just taking the product of the percents of each individual treatment effect. We're able to detect our true positives are the ones that look like they would respond in the assay, and they reach good clinical response, at least a 50% decrease in their disease. The true negatives are the ones that progressed or didn't reach that 50% goal, or likewise identified that they didn't get to a less than 50% combo effect. And so by this, we only had a couple false positive, false negatives, and we're able to compute that our assay was 96% sensitive for a partial response with just one false negative, and 86% specific for a partial response, just one false positive in that particular initial cohort. So I'm going to shift gears at this point to talk a little bit more about the antibody treatment specifically. So after we published this paper, we wanted to take some next steps to evaluate, can this improve how we're treating patients just in generally. One question that we often face is the potential to recycle anti-CD38 antibodies, which are very effective when they're first used in a patient, but we don't really know how well they work in a second line of treatment later on. With the immunomodulatory drugs and the protease inhibitors, we recycle them multiple times successfully, but these are newer drugs, so we wanted to evaluate this question. So the best one is the best target for this in multiple myeloma is CD38. It's a transmembrane protein that's highly expressed on myeloma cells, comparatively low on normal cells, on most normal cells. It has multiple roles in cell adhesion signaling, calcium mobilization. There are 2 monoclonal antibodies that I mentioned earlier that are FDA approved. Daratumumab was the first. Isatuximab was a later one. They both bind to slightly different epitopes, may confer slightly different activities, but have never been compared head to head. They work similarly like most monoclonal antibodies, rituximab being kind of the first, or Herceptin in breast cancer, in other cancers, through antibody dependent cellular cytotoxicity or ADCC. ABCP, or cellular antibody-dependent cellular phagocytosis, and the complement system. At least cetuximab has been implicated that it may also induce some direct apoptosis in cells. And in terms of a clinical trial for retreatment with this class, this one trial has been done, relatively small, where they took patients that had previously received daratumumab, gave them cetuximab monotherapy without a combination, and found that their clinical benefit was really minimal, and that one patient had a minimal response, and about half the patients had stable disease. Most of the patients that went into this went with basically from sequential lines of therapy, daratumumab right to cetuximab without a break in that therapy. So this is something that we thought we could study in kind of a more preclinical trial setting. So back to our MyDST paper, initially I found that daratumumab was quite active in the assay. So we're able to get a dose response curve that basically maxes out at 2 nanomolar, 2 or 20 nanomolar at the most. We look at kind of the waterfall plot here. So again, good responses are going down. Newly diagnosed patients or relapsed patients that they've never received daratumumab respond pretty well to this drug, whereas if they've gotten it before, they lose their sensitivity to it. So that's just shown again here in C. We see that the target goes down. So basically, the daratumumab resistant patients, they don't express much CD38 after they've received it once before. The daratumumab sensitive patients are the ones that really have much better CD38 expression. And so those two things inversely correlate. And kind of getting to that question we had before, this F is an experiment where we're adding something called collagenate liposomes, which is a reagent basically that you can buy that will poison the macrophages in the culture. So when we add the collagenate liposomes, we see that our sensitivity is, for the most part, completely blocked. So that macrophages are playing a big role in this culture effect that we see, similar to what they would be expected to do in patients. So I mentioned a preclinical trial. There's problems with clinical trials is that in multiple myeloma, we always have these huge clinical trials that take 3 drugs versus 2 drugs, and the 3-drug usually wins. So we have all this stuff that's approved, but it's probably likely better just because it's better to give 3-drug combinations in multiple myeloma than it is to give 2 drugs. So that kind of makes it a little bit less useful. And there's no head-to-head comparisons. So they also cost a lot of money, as you know, and they take a long time to read out, especially multiple myeloma. Like I said, in that first line setting, they can be in remission for 3 to 5 years. So there's some disadvantages to this that I think we could really help with preclinical trials using something like MyDST. So we took on this project really with the goal of, with the 2 goals that I show here, to evaluate what are the parameters that should dictate whether a patient is re-exposed to CD38 antibody retreatment. When is that going to be beneficial to the patient? And then likewise to compare daratumumab and cetuximab head-to-head in a way that we couldn't ever do in a clinical trial, and specifically in the retreatment setting here. So Olivia in my lab, now a resident in Washington, D.C., took basically MyDST results and started studying them from basically patients that had been daratumumab refractory and off of daratumumab for a period of time. So the x-axis here is the amount of months the patient's been off of daratumumab, anywhere from basically weeks off of daratumumab to out to 52 months off of daratumumab. And you can see that there's a correlation. Basically, you get more sensitive with your to monoclonal antibody retreatment, the ex vivo setting, with more time off of the treatment. So those people that have just gotten off of daratumumab or just a few months out, they're all pretty resistant. There's not much drug effect, if any, at this time point. When you've had more than a year, you start to see the results come back, and this is kind of a recapture of their response that improves over amount of time off of daratumumab. So if we break these into 3 groups, this is what your ex vivo sensitivity looks like in all our patients that we've tested to this point that are daratumumab naive. So it's about a 50% killing effect on average at 48 hours. Those people that are less than 12 months off of daratumumab, you can see that not many of them reach, basically none of them except for one here, maybe 2 have reached that 20% threshold of 20% killing. So there's not much killing going on in that group. And the people that have had more than 12 months or more than a year off of daratumumab, we can see that are resensitizing or starting to resensitize. They don't look as good as the naive patients, but they look better for sure. So we looked at CD38 expression to determine if that was the reason, and it seems to be explaining a lot of it. It's generally highly expressed in these patients that are naive. It comes down to a quite low level, and this is a ratio of the MFI, so the median fluorescence intensity on the myeloma cells divided by that on what we call non-plasma cells, all the normal cells that aren't myeloma, basically, so our normal cells. So goes down to a low level at the less than 12-month group, and then it starts to come back, doesn't get to as high probably as, again, as the naive patients, but it's improving. Interestingly, when we kind of see, is there a correlation between the viability in the MyDST and CD38 expression? There's not a correlation, but there's more of this threshold effect where you see them for the most part to need at least a 6-fold increase on your myeloma cells compared to the normal cells in the culture for sensitivity. And beyond that, there are other factors that are probably playing a role. It's like a 50-50 proposition at that point. So it's necessary, but not sufficient to get a response. When we looked at isatuximab and daratumumab, these are the results highly correlate. Basically, the antibodies behave quite similarly in this assay. So head-to-head comparison shows they're really near equivalent. At the same time, when we see significantly different results, which are shown by these asterisks, they usually favor isatuximab. So there's some implication that we see that if you've had a patient that's gotten daratumumab before, it's probably better to switch to isatuximab to get maybe a slightly better response. And maybe that comes down to the epitopes being slightly different. We've been looking at, I'll talk about this a little bit later, but can we measure this in peripheral blood so we can save the need to do a bone marrow biopsy, and found that we can do that. Again, this is similar results from peripheral blood showing that isatuximab and daratumumab they behave pretty similarly, but when we see the bars looking slightly better for one or the other, it's usually favoring isatuximab. In this case, none of those are statistically significant, but that's just kind of our impression over looking at a lot of samples over time. Now, I'll just bring in the rest of this. I didn't mean to animate this. So we, again, follow the people that we studied in this daratumumab retreatment or daratumumab refractory group. And I found basically a couple of interesting examples here. So in our MyDST, we see about, this kind of modest effect in this patient. So this is the assay result that we take when we take this patient's blood before treatment and after treatment, after one cycle. We see that clinically they also are basically, in terms of their peripheral blood myeloma population, it's decreasing quite nicely. Interestingly, or importantly, really, this patient has close to a minor response, but really just has stable disease. So you can see that in the past they responded well to their treatment, modestly. And at that point, when they're daratumumab refractory and we studied them, they're really pretty drug resistant, and they may have been sensitive when we took the sample, but they really don't have any really lasting benefits from it. So they were able to stay in stable disease for about 5 months. So we can see the live cells go down from the myeloma cells go down in their blood. Interestingly, we see that they really shift their CD38 expression very rapidly, though that goes down as well. You can kind of see that this nice, robust CD38 expression on the x-axis shifting to the left, even within 1 month of treatment. And then we're just kind of on the group as a whole, we had 8 patients that got retreated. Just their clinician decided to retreat them with various regimens. About the half the time they chose daratumumab-based therapy, which is abbreviated with Ds in this graph. And just, but most of them either they may have gotten a partial response at best, but they relapse pretty quickly. So it really doesn't have a lot of lasting effects. At best, it bridges them to another therapy. So when we look at the outcomes, about half of the people that we've studied have died at this point. The overall survival in the group is about 2.5 years. Those that were retreated and those were not retreated with anti-CD38 antibodies, there's no difference in their survival, which, I guess we were curious to see, would it be better, would it be worse? It's about the same. So indicating that of all the things we have available now for these patients in this kind of multi-drug resistant setting, it works about as well as everything else. And that includes CAR T cells and bispecific antibodies. Caveat is that it's a small group of about 30 patients. So that's kind of interesting. Now we had one patient that we were excited about from this group. This patient had it was daratumumab exposed but not refractory. And what I mean by that is they got an anti-CD38 antibody when they had this basically fourth relapse, responded well to it. And then instead of staying on it like we would normally clinically just leave them on it until they progress, so basically started a relapse and then continue until their next relapse, he went off of it. He went to an auto stem cell transplant and then went on maintenance with an immunomodulatory drug, eventually relapsed. And when that patient was re-exposed, when we studied them and they were re-exposed to anti-CD38 antibody, again, daratumumab in that setting, they responded much, much better than any of the other people in the cohort. So they got basically a complete response down to a normal free light chain expression. When we studied their blood at a second time point, we see that really all these myeloma cells that were in their blood are now gone. Maybe the ones that are still remaining have again shifted their CD38 down, but not as severely as we saw before. So provocative in that maybe we're dosing daratumumab or isatuximab for that matter all wrong by continuing it forever. And so we next asked the question, okay, what about a remission sample? Could we look at remission multiple myeloma and see if they have any CD38 that we could target? So what we're finding, and this is a little bit kind of in progress, but it doesn't look like CD38 is really targetable in these patients that we're continuing daratumumab or isatuximab indefinitely. So in this graph, we're using a different marker, CD46, because CD38 is downregulated. So CD138 and CD46, this identifies our multiple myeloma cells in this remission sample. So there are a few of them that we can measure. They express this other myeloma protein, BCMA, but their 38 is really at background level. And I don't think this is at all targetable based on these results. This is the normal cell graph in gray. It really doesn't express it beyond the normal cell level like it does in the patient shown in C who's never gotten daratumumab before. So again, myeloma cells identified in a remission sample, daratumumab naive, has BCMA expression and has really high levels of CD38, so kind of what we would expect. And so in a group of 6 people that are all kind of shown in B here in terms of being in remission, on daratumumab or isatuximab-based therapy, they don't express CD38 to any degree, and I would really expect we're just flushing drug down the toilet by treating these patients with an antibody at this point. They don't express the target anymore. So it's really fascinating, I think. So really kind of where we want to go with this next is a clinical trial where we test that, take a patient through an autologous stem cell transplant or basically anti-CD38 based induction, put them on maintenance therapy, and then wait to see how they do, and then retreat them with CD38 based treatment when they progress and see if our results are better. So that's all I'll tell. I'll finish up now. That's all I'll talk about with the anti-CD38 antibodies. I mentioned the blood, so we're trying to see if we can do MyDST 2.0, basically, where we use blood instead of bone marrow. And so we're studying bone marrow, matched bone marrow and blood samples. And what we're finding is that the myeloma is pretty usually present if they have a significant burden of disease. It's maybe about it's about tenfold or a log less than it was in the bone marrow. But we can still detect it and we can still measure it. We can measure the drug sensitivity of it in many people. And so we can kind of find that that these basically correlate pretty well. Lauren in our lab is trying to optimize that approach. So that's kind of our next iteration. This will open up our ability to do this test for many people without having to put them through a bone marrow biopsy, which is a painful procedure. We also have a prospective clinical trial open now. Peter Forsberg, one of my colleagues in the myeloma group, is the PI of this trial. So basically we'll take patients in the relapse setting. It's sponsored by Karyopharm, who owns Selinexor. So the Selinexor is the S in these triplets. So basically the decision will be, we're not using the assay to pick their treatments yet. Once we get, CLIA certified, if this looks good, we'll get CLIA certified and try and do this to actually pick their treatments. But they're going to get enrolled. We'll take a sample, and the investigator, or I should say, really their physician will make a treatment selection that's based on selinexor and dexamethasone. So either getting pomalidomide, which is a immunomodulatory drug, daratumumab, the anti-CD38, or Kyprolis, which is another, a next generation proteasome inhibitor, based on their clinical gestalt. And we'll test all these different drugs in MyDST, both in bone marrow and in blood, try and determine what kind of the best decision would have been based on lab results, and then compare how they did clinically with that result in a more prospective way. So this is now open, and we're about to start accruing patients to it. So we'll see how it goes. The other thing I'm really excited about is that bispecific antibodies are the next hot thing in multiple myeloma. So they look absolutely outstanding for patients with multi-drug resistant myeloma. So we started collaborating with different pharma companies, and this is the first one that we've gotten in our hands. It's basically an anti-CD38. It's a trispecific. It's targeting CD38 and CD3 like a normal bispecific, but this one is interesting that it also has a anti-CD28 binding moiety. So basically it's a T-cell engaging antibody. It bridges T-cells to multiple myeloma cells. And in MyDST, we can get a nice dose response relationship in this platform. And remarkably, it works at extremely low picomolar concentration. So basically, I guess this would be like 10 picomolar. We're starting to see activity and consistent up to 10 picomolar. So we're looking at screening patients at 1 nanomolar just to basically saturate our effect to get the maximum drug effect. Simultaneously, and Alana is doing this, a grad student in my lab is doing this work where we're kind of immune profiling T cells predominantly to determine, is it the number of T cells in the culture? Is it their activity? Is there a CD4 to CD8 ratio that's important, or what mediates kind of their response? So T cell activating treatments do work in this setting, so we're super excited about that. So I'll stop here and conclude with a few highlights. So we think that MyDST is going to be a useful platform for phenotypic personalized medicine and preclinical trials with drug companies on multiple myeloma. When we think about daratumumab or isatuximab, the anti-CD38 antibodies, we would recommend, based on this ex vivo results, that retreatment should not be attempted unless the patient's been off for at least 12 months. And there may be benefit from switching agents if that's easily clinically feasible. And I think that based on the kind of remission results that I showed in that one example, that the patient did much better when they weren't actually clinically refractory to daratumumab. They didn't relapse while they were on daratumumab. That this continuous treatment is counterproductive. It leads to stable downregulation of CD38 that's hard to recover from these cells, and that people maybe do better if we discontinue it after they get to a remission. Lastly, T-cell engaging therapies such as bispecifics can be measured with MyBST, and so we're currently trying to work on that and expand our repertoire of bispecific antibodies that we have to play with in the lab. And then we have this clinical trial open to kind of validate how MyBST looks going forward. So with that, I'd just like to thank the members of my lab, which Zach Walker has been with me in the lab since the beginning. So he's my lab manager and senior technician. Lauren Ryman is our new technician, who's working on MyBST 2.0. Olivia worked on the anti-CD38 project, and Alana Keller is our grad student. Alana and Lorraine are our grad students. Collaborators are Tomer Mark and Peter Forsberg in the myeloma program. I'm mentored here at the university by Craig Jordan and Clay Smith. And of course, we'd like to thank all of our patients who donate samples for this work, and our funders, the clinical team, of course. We couldn't do any of this without them. So thank you for your attention.